Okay, let's get started. Our clock in here gets slower and slower every day. So if I keep blowing by the time that we have somebody start to wave their hands around. So a few reminders, noon on Monday. Okay, so come at noon, don't come at 1:30, come at noon. You have an hour and 50 minutes to do the final. It is a longer exam than what we're used to. So, but I think that it will be doable. Remember, your homework is due at exam time on that time on Monday. I am going to tighten up ship a little bit. So I'm going to ask for the final that everybody put their backpacks off to the side. I need to be able to walk down the road. So everybody just keep all distractions away. And I'm also going to ask that no one leaves to go to the bathroom. I've had a few of you do that. I can't control what you do when you go to the bathroom, obviously. So go to the bathroom before you come in, and then it's reasonable that you shouldn't have to do that. So speak with me if that's a problem. But okay, so those two things. The exam, of course, you'll do the multiple choice on the Scantron, and then there's a matching portion which you will write on the exam, and I'll be hand grading that. Okay, so yeah, so there's a multiple choice that you'll put on the Scantron, and then the matching, there's 4 pages of matching. You'll do that right on the exam, and I will hand grade that later. So, okay, so that's a little different. I think that's the announcements that I had. Anybody other questions about the exam? Okay, I will be posting a few questions. I don't have a huge base of exam questions because I use a very similar exam with some tweaks every year. So I have a few that I can post if we don't get to doing any reviewing at the end of class today. So I will do that. I also have a little metabolism review that I'll post, just kind of, hey, remember what these pathways do type of thing. Okay. All right. So where we left off last time, hopefully you have a little bit of an appreciation that we don't have to deal with smallpox anymore. I think, let's see, I was trying to remember what we got to, what we didn't hear. Transmission, how is it spread? Mostly by droplets. Okay, so those kind of larger droplets when you're close to somebody, you know, speaking, coughing, sneezing. Less commonly, it can be airborne. It can be transmitted by fomites, some inanimate object, but that's going to be a lot less common. They think the R0, remember, that's a measure of how many people an infected person is likely to infect. I'll write that down here. The R0 is somewhere between 3.5 and 6. So we said measles was like, was it 15 or 18, something like that. Chickenpox was 10. So not as infectious as measles or chickenpox, but much more so than the flu. I guess you can put that in perspective. Remember there was 2 different, there was variola major and variola minor. That's the variola major that was more severe disease that was circulating before 1900. All right, so we started vaccinating in 1960s sometime. We'll talk about the vaccine in a second. And by 1980, it was declared eradicated. We stopped routine vaccination in 1972. So that means we would give the vaccination to kids when they were 1 year old. So roughly anybody born before 1971, so that's like 52 or older, 52 or younger has not had the vaccine. Okay, 52 or older has had it. And that's important as we've dealt with this mpox outbreak. If you've been vaccinated for smallpox, then that has a good deal of protection against mpox, or what we used to call monkeypox. Okay, so that's kind of the so what there. Just interesting, what was that? What did that vaccination look like? Thank goodness we don't have to do this anymore. They would use something called a bifurcated needle. And this looks primitive, but this wasn't actually an injection. What they do, they would dip this into the vaccine, the vaccinia virus. So you get a little droplet in there. And then if you were administering this vaccine to somebody, you would poke them, basically prick them with that bifurcated needle that had that drop of virus, attenuated virus in there. And you'd break the skin like 15 times. Okay, all within about 5 millimeters. So if you administered it correctly, then the patient would get this kind of big, angry blister, basically. So I can't imagine trying to do that to a 1-year-old kid. You have to tie them down, right? Injecting them is bad enough. So it hurt, and it was really sore. A lot of times patients had a fever as a result of that. You had to cover up that site of inoculation, because if you did it right, then the wound would shed live virus. So you had to cover it up so you didn't, accidentally come in contact with somebody that maybe was immunocompromised. But people could get the virus. They could get contact immunity just by coming in contact with you. Okay, so it wasn't a great vaccine, but people embraced it and we got rid of it, thankfully. So we said that vaccine was vaccinia virus. Again, lots of side effects, like fever, sometimes a rash, sometimes some complications just from the vaccine. So again, we don't routinely give that anymore, even though there is the worry that, we still have kept 2 stocks. There's only supposed to be 2 of smallpox virus, one at the CDC, one at a facility in Siberia. So there's actually, the worry that smallpox could be weaponized. So that's why we still have a stock of it in case, whatever, just in case. We will not be able to eradicate every infectious disease. What made smallpox a really good candidate to be eradicated? The pathogen had very little antigenic variation. So that means it didn't change. That DNA polymerase that smallpox virion used to replicate its DNA genome was really good. It didn't make very many mistakes. So unlike things like coronavirus, it's changing all the time. It had little antigenic variation, and just as important as that is that there is no animal reservoirs. Okay, so we didn't have the virus hiding out in some lesser bat or mouse somewhere, something like that. All right, so it's only passed around through people. And in addition, anybody that was infected, they are easy to recognize because of the rash. Okay, so you didn't have people walking around with asymptomatic infections. They started feeling sick and they started to be infectious when they got the rash. Okay, so again, hopefully we won't see that again, but mpox is a related infection. Of course, this is a disease formerly known as monkeypox. They changed the name just, I think it was just in like December of last year that they changed the name. Why did they change the name to mpox? To remove confusion about how it's transmitted and in an effort to reduce racist and stigmatizing language. All right, so that's why they changed it. What is interesting about mpox is that this has been around for a long time, kind of would show up every once in a while, mostly in Africa. usually when somebody was in contact with one of the animal reservoirs. So there are some species of monkey that are animal reservoirs, other things like Gambian pouched rats, tree squirrels, rope squirrels, certain species of mice. Okay, so not just monkeys. What's new is, so it always used to be people would get mpox when they came in contact with one of those animal reservoirs. What's new now is that they're still calling it an outbreak. They're not calling it an epidemic or pandemic. But now you have all these people getting it in all these new locations where we've never had mpox being transmitted before. So in terms of cases, I looked this up this week. We've had 30,000 cases in the US and 42 deaths. So, I mean, they typically say that the fatality rate for this is like 3 to 6% mortality, but we have not had that many deaths, certainly in the US. Worldwide, like 80,000 confirmed cases. And again, and most of those are from locations that have never had cases before. Worldwide immunity is declining to mpox. Again, that has to do with who's been vaccinated against smallpox. Anybody approximately 52 or older probably has immunity to mpox. So this is something that, even though the outbreak is subsiding now, this could show up periodically, certainly. What does the infection look like? Fever, intense headache, swollen lymph nodes. And that's after maybe a day or two of that, then the rash shows up. And it's kind of unusual to get a rash on the palms of your hands or soles of your feet, but that's one of the places that it shows up. And basically kind of spreads to lots of different places. Usually lasts maybe 2 to 4 weeks, and it's supposed to be pretty nasty. You can have milder cases, but patients feel pretty rotten. Again, 3 to 6% mortality is what they're saying, but it doesn't appear to be typically that high in the US anyway. This swollen lymph nodes, you know, a lot of infections cause rashes, but things like measles or chickenpox or even smallpox don't cause the swollen lymph nodes. So that's sort of a, I guess you would call it a sign and not a symptom. That's a little bit unique to mpox. All right, where we at with this outbreak? Again, you know, it's spread to lots of continents and it's higher than normal, but they're still calling it an outbreak. We're pretty much, it appears that we're past the worst of it. So this is from 2022, late summer and early fall. Again, cases have returned to being pretty low. Who's getting it and how are they getting it? We think there's some question about this to begin with, but we have to have really, really close, prolonged contact with somebody. So this is not airborne. The population of people, for some reason, it's mostly men that are getting it. I'm not sure if that's really understood why that is, but men having sex with men are a population that's had the worst transmission rates here. Again, just not because it's a sexually transmitted infection, but just because of that close contact. You know, in pockets in the country where transmission is high, they're encouraging people to get vaccinated. Okay, potentially indirectly by fomites, you know, like a blanket or sheets, but we think that that's not very common. All right, so we still have more to learn about that. Treatment, this drug, tecovirimet, that was just approved in 2004 to treat smallpox. So isn't that interesting that they went to the trouble to get FDA approval for a drug for an infection that we don't even have anymore? So they approved it, FDA approved it sort of emergency use, compassionate use for monkeypox, and it has some use against monkeypox. It inhibits the virus from leaving the host cell. And we didn't get too much into the particulars of that structure of that virion. But the vaccine, it's called by, there's several different vaccine, JNNEOS. I don't know if that's an acronym for something. It's basically that vaccinia virus that's been modified a little bit. So it's an attenuated vaccine that actually can't replicate. And there's no, poking, no bifurcated needles here. It's just an injection. So that's a good thing. And that's been approved both for monkeypox and smallpox. So we actually have like a couple million doses of that stockpiled, just in case smallpox would emerge. All right, so I guess the take-home cases are waning here, but that doesn't mean that it's not going to rebound or we're not going to see it again. Okay, questions about smallpox or mpox? Oops, the bottom here. All right, so we're going to finish off with a few different RNA viruses. So we had the herpes viridae and the orthopox viridae, or orthopox virus. No, orthopox viridae. I said it right the first time. As DNA viruses, and then several RNA viruses. I want to talk about flu, HIV, and if we have time, a little bit about COVID as well, just a little bit. All right, so one thing to sort of get straight, the infection influenza, probably you've all had it. A lot of times it's one of those things like nails on a chalkboard when somebody says to me, oh, I was throwing up all week, I had the flu. Like, okay, if you had the flu, if you had influenza, this is a respiratory virus. Symptoms from a respiratory virus are not typically throwing up. All right, so if you've got the stomach flu, we'll call it stomach flu, but regular influenza, it's respiratory, and rarely, sometimes kids throw up from that, but most of the time adults don't. Okay, so there's three kind of varieties of influenza that humans get: influenza A, influenza B, and influenza C. Okay, typically influenza A, those are the most serious ones. And that's the influenza that actually will cross species. So, swine and horses and different kind of birds can get influenza A. In influenza B tends to be less serious, although we do contain a couple of influenza B strains in our vaccine every year. Influenza C is usually pretty mild. Okay, so we don't worry as much about that. The B and C, again, don't infect other animals. They only infect humans. All right, so if we look at the virion here, what can we say about it? It's enveloped, just like herpes, just like smallpox. As far as the genome, it's negative sense single-stranded RNA. Okay, so that means it has to carry with them or have the ability to make the enzyme RNA-dependent RNA polymerase, RDRP, RNA-dependent RNA polymerase, in order to get the positive sense single-stranded RNA, which, remember, that gets read at the host ribosomes. Okay, so that's an enzyme that we never make RNA from RNA, so it has to have that, bring that enzyme with it. The genome is also segmented. Okay. And so you can see that in this picture here, it shows 8 different segments. Actually, influenza A has 8 segments and B and C have 7. Okay, so 7 or 8. And typically one segment will encode for one protein. Okay, so there's, you know, somewhere between 7 or 8 proteins that are made. Sometimes a segment will actually make 2. So kind of a many fewer genes. Remember, the herpes virus encodes for like 80 or 100 genes, smallpox 200 genes. So in general, RNA viruses have a much smaller genome. And it has a helical capsid. So I don't know, you can kind of picture that from that picture there. Spikes. On the outside of the virion, there are a couple different kinds of glycoprotein spikes. So in this figure over here, the red one is called hemagglutinin. Usually abbreviate that with an H. And the little one that they show with green in this picture is neuraminidase. And we abbreviate that N. Okay, so the hemagglutinin, we think that spike is most important for attachment to the host. We'll say attachment and fusion with the host cell. And the neuraminidase spike comes into play when the new virion is leaving the host cell. Okay, so it not prevents, we'll just say for release from host cell. Okay. And then influenza A, we name those subtypes of that virus based on what kind of variety of hemagglutinin and what variety of neuraminidase it has. So influenza A has 18 different hemagglutinin and 11 different. I had to update that. We've found a few more since last time I taught this class. So different varieties. So you've heard of probably H1N1 flu, swine flu. Okay, that's what the H is and the N's stand for, a different variety of hemagglutinin or neuraminidase. All right, so that kind of brings into, we have this naming rules for strains of influenza, and it's just sort of interesting. I think we can very easily understand what that nomenclature refers to. So here's a couple of examples down here. So that first A, that means it's an influenza A. Remember, that's the most serious ones. And then this is the host that it was first found in. So the host animal where it was found. And some strains don't include that. So if it doesn't include it, then you can assume it was first found in a human. All right, so swine, pig, equine, horse, the geographic, where we found it. And that's either a city, sometimes it's a state, sometimes it's a province, it's another country. This number is just going to be the strain number, the year we isolated it, and then again, the variety of the hemagglutinin or the neuraminidase. Okay. So I'll show the different strains that are included in this year's vaccine in a minute, and hopefully that will make a little bit more sense. All right, so it's those spikes that we kind of focus on. That's the most antigenic part of it's it's on the outside of the virion, so that's what our immune system is going to recognize. So we make a lot of fuss about those spikes, and influenza is pretty good at changing its spike proteins. If you remember back, the other pathogen that we talked about that was really good at antigenic variation was Neisseria gonorrhoeae. Okay, that changed its pili or fimbriae really, really quickly. Influenza is much more slow than that, and that variation happens a little different mechanism. There's two kinds of antigenic variation in flu: antigenic drift and antigenic shift. Okay, antigenic drift is when those proteins and the protein composition of the spikes just kind of changes by point mutation over time. Okay, so gradual changes in the amino acid composition of those spike proteins. The reason that we get those point mutations is because that RNA-dependent RNA polymerase tends to make a lot of mistakes. Okay, so we get a fairly high mutation rate. And we think that it's due to this antigenic shift that we get kind of every other year kind of outbreaks of worse flu. So we'll say biennial outbreaks. Okay, so if you see this graph over here, typically 1 year we won't have a problem with flu, and then the next year it'll be a little bit worse. So because of that, again, kind of slow change, we have a lot 1 year, and then the next year it hasn't changed very much. So there's not as much flu circulating, and it changes a little bit more. Oh, suddenly people don't have immunity, so we get more cases. So it's kind of an up and down thing due to antigenic drift. Now, when we see antigenic shift, then that's when we have typically a year where the flu is really bad. Antigenic shift is when we get a more abrupt change in those hemagglutinin and neuraminidase spikes, right? So let me come back to this in a minute. This graphic, it shows antigenic drift, kind of slow change over time. When we have an antigenic shift, these are in influenza A, only strains that infect other animals. And if you have, say, a strain of flu that only infects humans, here's a strain that only infects whatever, ducks or whatever, and you have two different strains infecting another host at the same time, like a pig, okay, then you get some genetic reassortment, and it's possible to have some novel virions. So this is the avian H3 and the human N2. So making something novel, and if it's infective to humans, transmissible to humans, then we've got a problem because our immune system doesn't have a very good response to that. Okay, so you can see why we're really closely monitoring influenza in parts of the world like China, where people live maybe in the same building with their pigs and their ducks and their chickens. There's a much greater chance that these kinds of reassortments are going to take place. So again, they really closely monitor, and the worry is that some really nasty strain is going to start in China. But with this antigenic shift, then we tend to have outbreaks about every 10 years of really bad flu. All right, so we've got the antigenic drift kind of every other year thing, and then you get a year again where maybe it's more transmissible or people just don't have very good immunity to it. The last time we had a really bad year was 2008. Right, so we are overdue. We're about every 10 years. So the last few years, there's really been a big push. Get vaccinated for flu, and of course also with the COVID has complicated things as well. Okay, so know the difference between the antigenic shift and antigenic drift. You all probably know what clinical influenza looks like. You probably had it at some point. It carries about a 2-day incubation. So you're exposed, and then on average, a couple days later is when you might start coming down with symptoms. It's respiratory, so they bind to the virions. You just breathe it in, and large droplets. We'll get to the transmission in just a minute. Host cells in that respiratory tract are being killed, and lots of inflammation results from that. So symptoms, high fever, chills, muscle pain, say muscle aches, cough, lethargy. And you know, when you have it good, it's a good 7 to 10 days. It's serious. You feel lousy. It's been a while since I've had full-blown influenza, but it's not fun. And then, of course, secondary infections are common. Your immune system is kind of either maybe knocked back a little bit or overactive in the wrong ways, and you're susceptible to getting ear infections or pneumonia. Transmission, large droplets. I think it has the potential to be airborne, but that's not commonly the case. For an enveloped virus, it's pretty hardy on inanimate objects. So also through fomites, okay, a doorknob, a computer keyboard. It's going to be viable for about a day on an inanimate object. And of course, it's going to be more easily transmitted in places where there's lots of people congregated together. So, you know, schools, jails, places like that. It's worth taking a minute to kind of think about the flu pandemic from 100 years ago. You know, flu to us, you know, there's actually quite a bit of mortality usually that can be associated with flu, mostly people that are immunocompromised or older people or very young people. But that flu pandemic 100 years ago was really quite a different kind of flu. They estimate anywhere from a third to half the population of the world was infected, and it had about a 5% mortality. So that's much higher than usual. About 50 million people died. That's kind of on the lower estimates. And of course, we have many more people in the world now than what they did 100 years ago. This was during World War I, and troops were moving all over the world. So of course, that didn't help. And they were actually, you know, in terms of treating it, are we better at treating flu now than what we were then? Well, oftentimes this flu would turn into pneumonia. And even though this was before we had antibiotics, they were pretty good at treating pneumonia because they would sensitize like horses to it and harvest antibodies from horses. And then they would treat people with pneumonia with antibodies. And they were pretty good at that. If you were overrun, like you see all these patients in a gym, you didn't have the capacity to do that. So they did have trouble treating the pneumonia. We would be better at that now. You know, we have some drugs to treat flu, but they're not very good. We're going to talk about that in a minute. This flu had a huge cytokine storm associated with it. So just out of control inflammation. So actually, one of the age groups that was the most vulnerable were people in their prime. So people your age up to about age 40, because they had too vigorous of an immune response. And they would have lots of fluid buildup in their lungs. They would often bleed, like from their nose, from their mouth. There were neurological consequences to this. So it was just kind of a whole different beast. And that's why we are just constantly monitoring flu and worrying about that. So yeah, here's a graph of, that flu pandemic, the people that were most affected. And again, you see this, 20 to 40 age group, which is really quite unusual. Treatment. All right, so we have several drugs that are called neuraminidase inhibitors. Okay, so that means they inhibit that spike neuraminidase. So they inhibit the release of virions from the host cell. Okay, so Tamiflu. There's a couple other ones, but Tamiflu is probably the one that you've most heard of. Oseltamivir is the kind of fancy way to say that. So it's but it's the brand name is Tamiflu. So here's a slide that kind of helps you understand how it works. So if you look at the neuraminidase activity, so the neuraminidase are this kind of brownish spikes in the virion here. And when that new virion has assembled in the host cell and it buds out from the host, so it takes some of the cell membrane from the host, and typically it will detach and then it will go on and infect another host cell. Well, if there's a presence of one of these drugs like Tamiflu, it's supposed to inhibit that neuraminidase activity. So basically, the new virions kind of stay stuck to the host cell, and then they can't go on and infect another cell. And if they do detach, the new virions typically will stay stuck together. And as we know, our immune system is better at noticing kind of clumps of things or large molecules. All right, so that's how that works. There's a new drug that has been approved. Let's see, I think it was 2018. Xofluza, and you might have seen commercials for this. I didn't notice them so much this year, but they used to say flu really sucks the life out of you or something like that. So that was sucks everything out of you. That was Xofluza. So this was a different mechanism. It's not a neuraminidase inhibitor. It actually inhibits these enzymes that act in cap snatching. All right, that sounds really weird, but we've actually talked about this. So if you remember, think back to when we talked about genetics, eukaryotic messenger RNA has on the 5' end, it has a guanine cap. And on the 3' end, it has a polyadenylation. Okay, lots of adenines. Okay, and there's exons and introns. We talked about that as well. But remember that transcript has these kind of protections on the end, because in eukaryotes, that messenger RNA has to travel from the nucleus, where it was transcribed from the DNA, out to the to the ribosomes in the cytoplasm. So it has this protection on each end to kind of protect it from being degraded out in the cytoplasm. Influenza virions, if they're going to be successful, remember influenza is negative single-stranded RNA. It makes itself into positive sense single-stranded RNA to get rid of the ribosomes. But remember this guanine cap, that is what docks at the small subunit of our ribosomes. Okay, so if the influenza virions, if their messenger RNA doesn't have that cap on it, it's not going to be recognized as easily at the ribosomes. So what the influenza virions do is they snatch that cap and put it on their messenger RNA. Okay. And then again, they can get their transcripts translated into viral proteins. So what this drug, Xofluza, does is inhibits that cap snatching. So the influenza variants have less chance of being translated. So it was a complicated explanation, but I wanted to go through it because remember, we talked about that before. All right, so what's the so what? If you compare these two drugs, Tamiflu and Xofluza, it's good to kind of think about it now, because when you do get sick, you might want to decide, well, should I take one of those drugs? Will it help? So if we compare them, of course, Tamiflu has been around a while. This was approved in 1999. Both these drugs, you have to give them within the first 2 days of symptoms. Why not after that? Well, in the first 2 days, after 2 days, you've got enough viral replication that a lot of the effects, the signs and symptoms we have are just due to our body's inflammatory reaction or the production of interferon. Actually, alpha and beta interferon causes us to feel lousy, basically. So when our immune system, when our cells are secreting that, that's what makes us feel bad and not necessarily the variants. So you got to give it right away just to kind of inhibit the replication of that variant early on. And both of them are by prescription. There's been a push to make Tamiflu over the counter, and I keep checking on that, but it isn't yet. So both of them, you have to go to a doctor. The Tamiflu, you have to take twice a day for 5 days. Interesting enough, Xofluza is 1 pill. So that's kind of easy. Tamiflu is generic now, so it's still somewhere between $20 and $50. Still more than a lot of drugs we have, but at least it's less than it was before because it's generic. And Xofluza, I just checked on this, it's somewhere between $150 and $180. So 1 pill. So it'll cost you, or it'll cost your insurance company. Even babies can take Tamiflu. I don't think they have tested it as rigorously as they want to for Xofluza because it's new. Last year they said 12 years and up. This year they've extended it to kids 5 years and up. So now they're saying it's safe for kids. There are side effects to both, though. The side effects of Tamiflu can be dizziness, nausea, vomiting, diarrhea. So not great side effects. I don't know. Do you just want to deal with the flu and not have to deal with the side effects? I don't know. They're saying that Xofluza has a little bit less incidence of side effects. So it has that going for it. Which one works better? Both of them, they're saying, will decrease flu by about 1 day. All right, so yeah, is it worth it? Is it worth the money? Is it worth going to the doctor? You got to weigh all these things. There's also, possibility of an allergic reaction. It's anecdotal, but I had a friend whose kid went into seizures after they took Tamiflu. So you have to weigh all that stuff. They do say the CDC recommends anybody that is immunocompromised or, maybe at higher risk of having worse flu. They do recommend that they take one of these drugs. For people that are otherwise healthy, it's probably not necessary. It's probably not worth the trouble. But again, everybody kind of has to make their own decision there. All right, so we don't have great drugs. What about a vaccine? Well, due to the antigenic variation, typically the vaccine is somewhere between 40 to 60% effective. So it's not great. We have to kind of guess on the formulation of the vaccine by February. So they met a month ago to decide the formulation for next year. So we have to decide way ahead of time because most of our flu vaccine is grown up in eggs and it takes a while to grow that much up. There is a lot of different kind of formulations of flu vaccine, but the one that most people get, it's an inactivated virus. So again, they grow these up in eggs, chicken eggs, and then they kill it. And they make sure that the vaccine has been inactivated. There is an attenuated form that you breathe in through your nose. This tends to work best for kids. I think it's between like the ages of 8 and 12, something like that. It's again a weakened virion that will only replicate in like the cooler areas of the nose. So it has some kind of, they get kind of a mild cold-like symptoms. Okay, so you know, in some years we get good protection. Some years not as good because it's mutated and, you know, included in the formulation, you know, it's just changed too much from that. But you cannot get flu from the flu vaccine. You probably all heard somebody say that. Again, it's an inactivated virus. You can feel kind of lousy after getting a flu shot. I usually have, my arm is really sore, at least for a day. You know, sometimes people have a little fever, less commonly, but it's not the flu. Unless you happen to, you know, it takes a couple of weeks for it to start working to get the memory cells beat up, kind of made. So you could actually, you know, be exposed to flu and get it, but not from the vaccine. And interesting, so I put this year's formulation next to the one for the following year. Interestingly, the strains are, everything is almost the same, which is unusual. Oftentimes they'll include like a totally different strain. Both of these strains were isolated in Australia. Victoria and Darwin are cities in Australia. And the only difference is that this one is a little bit different strain than the one that we used last year. They almost always include an H1N1 PDM09, that stands for pandemic, like the one from 2009, 2008, 2009. That was a very severe year, so I almost always include one that's similar to that. All right, any questions about flu, about vaccine, about treating it? Okay. All right, yes.
How do they determine which strains they're going to use? So you said they choose like one that was from a recent pandemic, but outside of that,
how do they choose? There's a whole committee that surveils everywhere. They're especially looking closely at what's going on in the Southern Hemisphere during their winter, and see what was circulating there, and then try to kind of project what's going to be circulating here. So there's people that's their job. But yeah, they don't always get it right. All right, so let's move on, talk a little bit about HIV. So a family Retroviridae lentivirus is the genus. That's a little bit less recognizable, possibly. If we look at the structure of this virion, once again, it's enveloped. It has a segmented plus sense strands of RNA. So it's actually two different segments. You can see that here and here, two identical. Oh, did I bump something again? Let's see. Okay, sorry. I very nearly forgot my iPad today, so at least I didn't do that. Okay. Back. No, don't see me. Let's try this again. Okay. Come on. There we go. Good. Oh, okay. So what I was saying, there's two segments that are copies of each other of plus sense single-stranded RNA. Key enzymes. There's also in this, I really like this little figure here. There's these dots right here and here and here and here. Those are proteins. Virion also carries with it 3 proteins that are really important for it to reproduce itself. One is reverse transcriptase. Okay, we've talked about this enzyme before. So it takes this single-stranded RNA, and instead of when they first discovered this enzyme, it was one of those really like mind-blowing things because it goes against the dogma of the passage of information the way it happens in our cells. This virion can take single-stranded RNA and make DNA from that. So it goes to single-stranded RNA to single-stranded DNA to double-stranded DNA. Okay, and reverse transcriptase catalyzes all of that. So that's something, remember, that we don't do in our cells. We make RNA from DNA, not the other way around. Okay, another key enzyme, integrase. I guess I should say, why would a virion do that? Like, why when it's already plus sense single-stranded RNA, why doesn't it just go read that at the ribosomes and call it good? Why does it go back through a DNA intermediate? Well, DNA is really stable, so maybe that's one of the reasons, but also if it makes DNA, then it can integrate into our host chromosome and hide out there. So in addition to herpes virus, HIV is one virion that can go latent. Okay, remember that lysogeny in animal cells we call latency. So this enzyme integrase will enable that DNA, enables viral DNA to, we'll say integrate, for lack of a better word, into the host chromosome. And another enzyme, protease, that's necessary, we'll say, for maturation of the virion. We're going to talk, if we have time, about COVID. It has a protease as well that's necessary. Works a little bit different way, but to make some cuts and some enzymes so that they're functional. Some key glycoproteins in the surface here, GP120 and GP41. So GP120 is the kind of head of that spike protein that's necessary to recognize certain receptors on the host cell. And then kind of the bottom of that kind of spike is necessary once that virion enters into the host cell to kind of catalyze the uncoding of it. All right, so I like this graphic. It kind of shows the life cycle of the virion. There's actually some disagreement as to how HIV actually enters the host cell, whether it does membrane fusion or endocytosis. They show it entering by endocytosis. Okay, it's receptor mediated. So it's recognizing the CD4 glycoprotein that are on the outside of some of our cells. Of course, we associate that with being on T helper cells, but it's actually on other types of cells that we didn't really talk about as well, like macrophages. So HIV can infect lots of different cells besides helper cells, but it really causes the most harm to T helper cells. All right, so that GP 120 again helps the virion recognize the right host cells and catalyzes however it enters in the host cell, and that GP 41 causes the uncoding of that endosome and the viral envelope and the capsid. And what we have spilling out here is that viral single-stranded single-stranded RNA. Because it carries with it some reverse transcriptase, that RNA is made into double-stranded DNA, and then due to the integrase, that gets inserted into the human DNA. So we call that the provirus. Remember, that was prophage, during lysogeny for bacteriophages. We call it the provirus there when it's inserted into human DNA. When that virus gets transcribed and translated into the new virion, that's kind of what's showing here. And then that protease functions somewhere. It might be before the virion leaves the cell. It might be once it does leave. I've seen it drawn differently, but there has to be some cuts made in certain proteins so it's a viable virion. All right. So it's those three enzymes that we've kind of focused on in the treatment. So let's talk about that treatment next. About half of the drugs we have to treat HIV are some kind of reverse transcriptase inhibitor. Some of them are nucleoside analogs. Remember, that's a compound that looks very similar enough to a nucleoside that it gets incorporated into the viral DNA. AZT was the very first drug that we had. So here's the structure of that. Here's the thymine base. The deoxyribose is very similar, except it's got this azide group here instead of a hydroxyl group. So when the virion incorporates that nucleoside into its DNA during replication, this will cause chain termination. Okay, that no more nucleotides can be added on there. And there are other reverse transcriptase inhibitors that work just by binding to reverse transcriptase and keeping it from working. There's drugs that act to inhibit the integration of HIV into our chromosome, and also ones that inhibit that maturation that have to take place. So we have a lot of different drugs now. Unfortunately, they all have at least possible side effects. And if I were to list all the side effects, they would take up this entire slide. Like really almost anything you can think of, not only short-term things like kind of nausea, fever, tiredness, but kind of long-term things like cancer and kidney shutdown, like really almost anything that you can think of. The good news is that usually the side effects are the worst when a patient first starts taking the drugs. So the first couple of weeks, and if they continue having a side effect that's not tolerated, then they can usually switch and try a different drug. Usually they're taking kind of a cocktail of, several different drugs to make sure that viral replication is stopped. Patients have to take these drugs their whole life. As soon as they stop, then the virus can start replicating again. Okay, so again, usually they can switch and find something that they tolerate pretty well. Pretty pricey. I just looked this up, anywhere from $20,000 to $50,000 a year to be on these drugs. So yes, we're getting better at treating it. People are living close to normal lifespan now, as long as they stay on the drugs. People that are HIV positive. Transmission, direct, specific ways. So blood transfusion, sexual intercourse, dirty needles, mother to child. About a third of children born to HIV positive mothers are HIV positive. So it's not a sure thing, but a pretty common thing. There's not any fomite transmission. It's really wimpy. The virion's really wimpy in the environment. You can't get it from a toilet seat or something like that. And it's not, of course, it's not airborne. So very direct means. Still a huge problem. 37 million infected worldwide. I haven't checked my numbers. Let's see. We're still having over 1.5 million infections a year, and about 700,000 deaths a year worldwide. So we're still more AIDS deaths than malaria, to kind of put that in perspective. But again, as long as you stay on the drugs, life expectancy in 2011, it was 70 years. Today, again, it's pretty much normal. In 1996, for somebody that was 20 years old that was found to be HIV positive, they had about 10 years to live. So we've come a long way. Okay. If you look at the pathogenesis of the disease, this is kind of a complicated looking chart, but it's helpful in terms of we've talked about all this stuff. Okay, so the x-axis here is time. This side is the y-axis here is talking about the CD4 count, CD4 helper T cell count, and this side is the number of HIV virion particles in the blood. So if you try to follow how much HIV is in the blood and the number of helper T cells and the antibodies that we're producing, all these things are related. So when a patient is first infected, they get a big spike of HIV in the blood. And of course, that's impacting our CD4 helper T cells. So their numbers are going down, but you have enough of them to begin with to kind of, be able to activate both B cells and T cells. And you do get the virus titer to go down a little bit. So HIV in the blood, again, initially we have some response at least. And then oftentimes, when it's a provirus, it's integrated, it will go latent for who knows how long. It kind of just sits there and hangs out and gets replicated every time the cell gets replicated. But eventually it will kind of reactivate and you'll get the number of virus particles as it's rising, the number of helper T cells is going down. And so again, those helper T cells can't activate B cells and the number of antibodies is going down. So as the virus titer increases, a person is considered to have full-blown acquired immunodeficiency, full-blown AIDS, when they have a CD4 count below, I think it's either 200 or 300 cells per mil. And eventually patients die due to other infections, things that typically wouldn't make them sick, opportunistic infections. So what kind of opportunistic infections? I found this list kind of helpful. I thought maybe this one's kind of old. I got this from the CDC. I tried to find a little different one, but no matter what your CD4 count is, if you have one or more of these opportunistic infections and you're HIV positive, then you're considered to have AIDS. So some of these things you'll recognize, some of them not. A few of these infections are things that anybody can get, like TB or coccidiomycosis. We talked about that a long time ago. That's that fungal infection that you can get from breathing in the fungal spores. But a lot of these are, things like septic salmonella. Okay, anybody with an any kind of immune response is not going to have problems with that, or particularly invasive herpes simplex infections. Again, unusual things that you don't normally see in people with a reasonably immune response. Okay. Any questions about HIV or AIDS? Encephaly, HIV related. Encephalitis is just inflammation of the brain. So I don't know exactly, kind of sounds similar to meningitis to me, but I actually don't know what the difference is there. So good question. And that was one of the things, like, back in 1980, when patients started showing up with some of these infections, doctors were like, well, how is this? Why are people, getting these infections? And that's when they started to figure out something else up. All right. Nobody wants to talk about COVID anymore. Everybody's tired of it. I want to just mention a few things about it, maybe something that you didn't know before. Look at the structure of the virion, talk about what we are doing now to treat it. And also some related viruses. So clinically interesting members of this Coronaviridae family or coronavirus genus. The name of these are the name of the viruses here, and then the infection that it causes. So SARS-CoV, that burst out onto the scene in about, it was about 2003, might have been 2001, I'm forgetting. But that stands for severe acute respiratory syndrome. And that first showed up in China, hopped on a plane to Toronto, a couple other places, Vietnam. And that was a very, it was related to COVID, but it was a much worse infection, a higher mortality rate, and it wasn't as infectious. So we were able, although there was maybe 2,000 to 3,000 cases, we were able to stop it. And there hasn't been any new cases of that since 2003. So that was what we were hoping would happen with SARS-CoV-2, but it didn't work out that way. MERS, that stands for Middle East Respiratory Syndrome. And that carries about a 30% mortality. So that is a much more serious infection. And that is, I think, that showed up a little bit later, maybe 2012. That's ongoing. We still have cases of that, not a lot, mostly in the Middle East, because people get it when they've been around camels. Okay, so the camels are the reservoir. So they have a lot of rules now about how you can handle camels, and you have to have personal protective equipment on. And they're working on a vaccine to give to camels so they don't transmit it to us. But again, that's pretty serious. We're not likely to come in contact with that here. And then, of course, SARS-CoV-2, that causes the infection known as COVID-19. When we looked at the genome of the spike protein of this new virus, it was very similar to SARS-CoV. That's why we named it SARS-CoV-2. Okay, these are all plus sense single-stranded RNA virions. They're all enveloped. Okay, if you look at the genome, it has an unsegmented genome. So that means it's all in one strand. So that's what's kind of drawn here with the little beads around it are supposed to be the capsid proteins. It actually, coronavirus has the longest, we'll say largest genome of any RNA virus. It encodes for, we think, 29 proteins. So that's kind of interesting. Helical capsid, the structural proteins, of course, it has the S, that stands for the spike protein. That's the one that all the vaccines are against. That's the most antigenic part of the virion. That's what it needs in order to bind to our host cells, those ACE2 receptors that we have. There's a couple of other things here. This envelope protein, that's necessary for assembly and release. Okay, we could mention some others, but I don't think we'll go into it. So just some structural things. All right, so I won't get into reservoirs or anything like that, because we don't really understand that very well for at least COVID-19. Originally, we thought it was probably a fruit bat, which the SARS-CoV-1 was the reservoir, but a lot of things are pointing to, we can't really find traces of this in nature. So that's why a lot of things point to the Wuhan Institute of Virology in China. They were doing studies, they call it gain of function, thank you, gain of function studies, where they were trying to take a coronavirus that was not infective and try to make it infective. So there's an argument that nobody should be doing gain of function studies. I don't think we'll ever know for sure whether it came from there or not, but there is some evidence there. So we won't worry all about that. You all know the signs and symptoms. It kind of depends on the variety of COVID, the strain that you get, whether it was Alpha or Beta or Omicron or whatever, what kind of symptoms you have. The newest Omicron variant, which is just starting to show up on the news, it's X something. Yeah, XBB 1.16. That's just starting to make the news. It has a new symptom. Has anybody heard it? So exciting, conjunctivitis. So like pink eye. Great. Yeah, so in addition to all the other stuff. And I always whenever I teach this, I put down, the deaths so far in the US, we're way slowing down. So that's the good news. Everything, hospitalizations, people that are infected, deaths, everything is trending down. So even though, we've got this new variant that we're kind of looking at, we don't think it's any more transmissible or any more severe. So that's the good news. Of course, transmissible by respiratory droplets, probably less commonly airborne, less commonly by fomites, but mostly those large droplets that would be caught if you had a mask on. So that's why we wore masks for so long. Okay, let's see. Treatment. Let's move on to that. We didn't really talk much about vaccines. Of course, RNA, DNA, virus vaccines. I don't want to really go back into that. The treatment, of course, we were trying, ventilators. We tried monoclonal antibodies. The trouble with the monoclonal antibodies, these are antibodies that are made in the lab. The trouble is, again, it's just changing enough that once we get some monoclonal antibodies, they're not very effective. So we're kind of, what I'm reading, they're kind of giving up on those. It had a few nucleoside analogs. Remdesivir was a drug that was already approved for use in Ebola. They found that to be not much use for COVID. So they studied that pretty carefully now. They were really excited about molnupiravir. That just came out about a year ago. It was the first drug that could be given by pill form. It's a nucleoside analog, but it worked a little bit differently. It wasn't a chain terminator. It caused catastrophic mutations. So basically, this nucleoside would get incorporated into the DNA, but then when that DNA was used as a template for replication, it could bind with pretty much any base. So it caused these catastrophic mutations. This was only very narrowly approved by the FDA because it was argued, do we really want COVID-19 to mutate more? Maybe not. So luckily, about the same time, Paxlovid was approved. It's a different mechanism. It's a protease inhibitor. And if you're interested in this, this is a really nice animation that Pfizer put out that shows exactly how it works. And here's another article that kind of talks about the drug a little bit more. It's about a year old. But this protease is an enzyme that has to,